Welcome to Dr. Balaga's God Knowledge Doc podcast, your quick dive into the latest medical breakthroughs to keep you ahead of the curve. Let's get started. Today is 16 June 2025. This video is on a Nature news article regarding tiny human hearts grown in pig embryos for the first time. The heart started to beat in the pig-human hybrids, which survived for 21 days. This was presented at the annual meeting of the International Society for Stem Cell Research in Hong Kong. Lai Lingshui, a developmental biologist at the Chinese Academy of Sciences, and her group presented their data. They knocked out pig genes for heart development and injected human stem cells. Embryos developed for 21 days. Hearts began beating and contained fluorescent human cells. The humanized hearts were fingertip sized, cells visibly integrated but not quantified. The study was not peer reviewed, unclear integration depth and immune interaction potential. Offers promise for bioengineered transplant organs with technical and ethical hurdles ahead. The second article was published today, 16 June 2025, in Nature, again from the same meeting, titled Mice with Human Cells Developed Using Game-Changing Technique. The human cells were injected into amniotic fluid, find their way into fetal mouse organs. It was presented at the International Society for Stem Cell Research meeting in Hong Kong on 12 June. Xiling Shen, a biomedical engineer at the UT Texas MD Anderson Cancer Center in Houston, and Huang, a developmental biologist at Terasaki Institute for Biomedical Innovation at Los Angeles, led this effort. Human gut, liver, and brain organoids were injected in the amniotic fluid of pregnant mice. These organoids self-targeted corresponding organs, the gut, the liver, and the brain, without breaching embryonic walls. 10% of the mice had about 1% of human intestinal cells, lower presence in the liver and cortex. Human cells were functional. Example, liver cells made albumin and persisted for 2 months. This again is considered game-changing for creating targeted humanized animal models. Ethical frontiers. Human cell integration into brains raises concerns about cognition and identity. There's a need for strict guidelines on Chimaera research boundaries. Scientific limitations are also there. Low human cell chimerism, less than 1%, limits functional organ use. The lack of peer-reviewed data undermines reproducibility and verification. What are the future directions? Improve cell targeting, integration, and immune compatibility. Require transparency, ethical oversight, and long-term functional validation. And these are vital. If you enjoyed this episode, share it with your colleagues. When we change how we see disease, we change how we shape lives. Until next time, stay curious, stay compassionate, and keep seeking wisdom. Podcast brought to you by Master MedFacts at mastermedfacts.com.